Boys and girls, a philosophical question has come to mind. Why are we doing the weather report for July 5th, 2022?
Indeed. It's Tuesday, isn't it?
Yeah, it is. Why are we doing it? And I have half a voice today, so I'm not sure how long this is going to go.
Yeah. So everyone in chat so far, give a thumbs up for Speed's voice. Please.
Yeah, it's going. It's going. I don't know where it's going. I think it was from watching Stranger Things.
That might be it.
I almost swallowed my tongue a couple times. So anyway, I'm not sure.
That's a neat trick.
Yeah, it is. Yep, getting it back up is the real trick. Swallowing it, that's the easy part. I do that all the time. Anyway, so we got a couple of things. So I did a little short video that just showed John Maddox resigning, and and he asked, are you going to double down on this 9% thing?
Yep.
Yep, we're going to double down on it.
Oh, thanks, YouTube. Yeah, I know now.
What's that?
This Answers in Atheism is going live. Shocker.
Oh, yeah. Oh, okay. Oh, God, that's wonderful to know. It tells me that, too, on my phone because I have a different account. Let me see if I can
it does that a few times. It's so funny.
Yep. Heretic Grower. Let's see. What do we want? The Deeper Genome. Okay, so the book is The Deeper Genome. And where is that book? So what happened is John Parrington wrote a pretty damn good book on the jungle of the genome and all the different, things that we're discovering about the complexity of the genome and all this stuff.
All
the new things.
But it's not made of wood or concrete. Yeah. It's made of molecules. Yeah.
We've been discovering things for over 20 years, that have been surprising. No, nobody's really surprised anymore when we discover some new, incredible little twisty thing going on in the genome. But anyway, let's see. So where did he write this 9% thing? I'm trying to figure out what page. Oh, okay. So on page 94, this Parrington wrote, but if only 9% of the genome is conserved in this way, okay, well, up here, let's see. Oh, he said, but if the whole genome is compared to that of the mouse, the similarity is far less, only around 5%. Now, he doesn't reference any paper for that claim, and I can't find a paper that makes that claim. In fact, I don't think anybody's actually done what you would have to do in order to come up with a number like that.
Right.
And that is going to take a little bit of unpacking, which we'll probably do a little bit later on, as to what you would have to do. So what they normally do is they look at the conserved regions, basically the regions that are pretty homologous, and they come up with a percentage difference between those regions. And Maddox's complaint was, oh, well, that's just the proteins. Well, okay, ultimately in the cell, it is the proteins that do the end job. I mean, it's pretty hard to deny.
Yeah.
I mean, no matter if it's mRNA, if it's a 4D nucleome, the arrangement of the chromatin or whatever it is, ultimately the purpose is to create a proteome, and that is a certain percentage of different proteins in that cell, and across that cell. That's what happens. Now, I guess to some degree you could say, well, signaling sequences are going to affect where the proteins go in the cell. So there would be that. But that's in the gene itself also. So it's the genes that they're comparing all the time. And
right. And it's not to say that we can't compare non-gene sequences occasionally. It's just a lot more difficult sometimes.
Yeah. And then in rats and mice, they have these B1 and B2, I think, or B1 and B3 signs that are inserting actively in the rat and mouse genomes. And in humans, we have L1, and we have all these signs and lines and ERVs that inserted into the primate sequences after we split from rats and mice, somewhat 71, 91 million years ago, that sort of thing. So it was a long time ago. Okay, so anyway, 48% of our genome is lines, signs, and ERVs. Oh my! So right away, 48% probably isn't going to compare too well. So you have to cut those out because those are transposons inserting. And of course, the creationists can claim that they're all functional, that they all had to be in that spot because God put them there. Yeah. So they can claim that, but then they're going to have to prove that to us for every one of them. But anyway, so then you're left with like 52%. And then what you have to do is you have to consider that there's 28% of our genome is introns, the area between exons, which are the actual coding part. And in the introns, we found some functionality, but we haven't found a hell of a lot. A lot of that stuff ends up on the cutting room floor after the gene is transcribed in the nucleus. Then it is spliced together to form the mRNA that leaves the nucleus and becomes a protein. Okay, so we have all these little snippets of introns that are on the cutting room floor, and some of those, granted, have small RNAs in them, and even some long non-coding RNAs that have some effect on the overall functionality. But we found like handfuls of those in a sea of introns. So these introns are normally marked, and they're normally degraded within 6 minutes of transcription. They have a half-life of 6 minutes in the nucleus, and they get turned back into raw nucleotides, and then they can be reused, right? Yeah. So they're used over again. Seems wasteful, but there are a lot of good evolutionary reasons for introns to be in there. But there's a hell of a lot of intronic space. When you look at any gene, you're going to see these little green lines, and you're going to see them in the middle of a sea of intronic material that ends up on the cutting room floor, that sort of thing. So anyway, so you would have to take the intron of each gene, and you would have to compare it to the intron of the similar gene in the mouse, okay, or the rat.
Right.
But then you would have to cut out the 5 or 6 lines and signs and ERVs that are stuck in the middle of that because of transposition, and then try to align the rest of it. And the truth is that nobody has ever done this. So that 5% figure he came up with, I think, is bullshit. I think he made a mistake in his writing. What do you think?
Right. Yeah, or just maybe didn't clarify it enough, perhaps, or I don't know. We can only guess at this point, but
yeah, because later on he talks about conserved regions.
Right. So he obviously knows what that topic is too. So
right.
I mean, with the context of the page in question, it seems to me like he was kind of talking. He expected the audience or the reader to know the context there, that it was talking about conserved or more properly constrained regions.
Well, yeah. What he was doing is he was explaining why. Okay, Dan Grar, who had a real problem.
He was responding to something Grar said.
Yeah, and Grar was saying, well, if only 9% of the genome is conserved, and he was okay in that statement. He says, but if only 9% is conserved, so he says, but if only 9% of the genome is conserved in this way, then by definition, only it can be functional. Otherwise, the conservation of the sequence would be much greater. Well, I don't know if that's a true statement either, because some of it could be functional in making the rat a rat and the human a human.
Yeah, that's true.
Yeah. But now the creationists and Faz Rana too, for some reason, are maintaining that all of these little differences that makes a rat and a human take the entire genome to do that for some reason. It takes 100% of the genome to turn an embryo into a rat or an embryo into a human. Okay, right. That's what they're claiming. In reality, it is a very, very, very small, tiny, probably 0.1% of the genome that's involved in what becomes the actual body plan and structure in embryos. Because the algorithm is a lot simpler. It doesn't need little tiny changes across an entire genome to make a rat or a human.
Yeah.
It's not that many modifications. And that's what I mean by it. The algorithm is a lot simpler than what it produces. It produces a very complex thing.
Right.
But it takes, like if you change the regulation of one gene by 20%, you'll end up with a limb that's 5 times as long.
Right. Well, it also depends on what gene you're looking at in particular, because if you're looking at a gene that regulates other genes, so to speak, then you can end up with some a bunch of effects down the line.
Right. They call them cassettes, basically. Yeah. And Hox genes are like cassettes.
Yeah.
And Hox genes turn on big cassettes of different proteins, and that can make huge changes. So a little tiny tail wags the whole dog. But they're claiming that for some reason God had to spread this information across the entire genome, and every bit of it has an effect. And I think the reason they think that is because they're still naively thinking of the genome as a blueprint for how to build a body, right?
Right.
Like you got to have, for the pinky, you got to have one blueprint.
Right.
That's going to take 5% of the genome. And then, for the middle finger is going to take a lot of the genome.
Right.
And the thumb somewhat less. But anyway, that is naive thinking. That isn't how embryology works. So anyway.
Yeah, so but the point you're trying to make here, just to wrap it up, is that when geneticists and in phylogenomics, you're not just comparing a 1-to-1 ratio of, you're aligning these sequences and you get this straightforward answer of it's this percentage of similarity. What you're saying is it's a lot more complicated than that, obviously, right. Because you're looking at different regions with different selection pressures and different constraints in terms of how they can mutate, for example. And you have to take all of that into account. And so it's more commonly, or we're comparing this subset of the genome to this subset of the genome, and it's this percent similar.
Yep. Decil Drace is going to put you in the corner.
Yeah, I know. I saw that email a while back. I forgot what what he's talking about specifically. I think it was the genetic code paper. I don't know which paper I brought up specifically. If you could elaborate a little, Decil.
Was it the Sutherland video? Because Sutherland hasn't posted the paper yet.
I have no idea, to be honest.
Oh no! So you're gonna have to send another email, Decil, and remind Andrew.
I know what email he sent me. I need to go back and look at it later.
Okay. Gotcha. Okay, so anyways, there's a little article here in nature.com, SciTable Knowledge Library. They have a whole bunch of stuff, a whole bunch of educational information. But I thought it was kind of cool. The comparisons highlight some striking findings. For example, while the tiny flowering plant, Arabidopsis thaliana, has a smaller genome than the fruit fly, Drosophila melanogaster, if you want to say things funny, 157 versus 165, yet it possesses nearly twice as many genes, 25,000 instead of 13,000. And then when you look, like Dr. Dan mentioned, some amoeba that has like 16 times as many nucleotides as we do, and then there's the onion, and there's there's no relationship to genome size,
right?
And how many genes there are, for one thing, and what kind of organism it produces as far as complexity. So So anyway.
Oh, okay. That paper. Yeah. So I know what you're talking about. It might take me a little bit to find that again because I don't remember which paper I used specifically. So.
And when you're at it, copy me on that paper.
Okay.
This too. Anyway. So anyway, it's a pretty interesting article, and right here, I'm not sure if I understand it. This illustration over here shows conserved segments of the human and mouse genome. And what they found, like for rats particularly, they found something like 230 to 260 huge sections of the genome that are like 40 million base pairs or more that are synchronous. That is, the genes are lined up the same and they have a strong homology. And because of this, they've been able to use the rat to do a lot of medical testing for humans.
Right.
Because there's that much similarity in the biochemistry of humans, which is kind of interesting. So
yeah,
anyway.
I kind of I don't know, but I kind of doubt it. If we were only 9% genetically similar overall in total to a mouse, how would we be able to have all these medical breakthroughs based on mice? Like that just doesn't make sense to me logically.
The bottom line is, if somebody says as well, we're only 9% similar if you count the whole genome, number one, they're trying to sell you something. Number two, they don't really know what they're talking about.
Right.
And number three, if they do know what they're talking about, then they're lying to you. They're cheating you. Right. So, because it's not that goddamn simple.
Right. Yeah. So LPP, unfortunately, has to be one of those three.
Yeah.
Sorry, folks.
And the thing with Faz Rana was kind of interesting because they got into junk DNA, because junk DNA is the same kind of conversation, right?
Right. Like, or real quick, should we maybe kind of elaborate the difference between just sequences being conserved or similar versus being constrained due to like purifying selection, which is what that 9% number is about?
Yes, because they get into that in this video.
Oh, okay. That I'll wait on that.
Well, no, I'm not sure if I'm going to be able to play that part of the video, so we can talk about it.
Oh, okay.
Okay, so conserved sequences would be things that are really like, okay, if you're looking between primate and primate, chimpanzee and human, right? You're going to find a whole huge section of the of the genome that is the same or conserved between those two, and those obviously have something to do with the making of a primate and the running of the biochemistry of a primate. Okay. But then when you look between like the rat and the human, you're going to find less, because there are going to be a few differences in the biochemistry, and there's going to be quite a few differences in the structure of the organism, right?
Right.
Okay. And let's see, but we have to elaborate on the difference between purifying and positive selection of genes. And
right.
Yeah. So can you take over?
Yeah. So as far as I understand it, positive selection basically is where there's a direct benefit of some mutation or DNA difference, such that it becomes more common in the population. It gets fixed more easily in the population over time.
Yep.
So it become more common in the genomes of whatever organisms you're looking at, respectively. Whereas purifying selection is basically, it removes deleterious mutations. So those kinds of mutations or regions tend to be less mutated by these deleterious mutations than expected, if they were just kind of doing nothing, basically. So like if these regions are biochemically important, and if a deleterious mutation or a mutation in general happens to land there, is more likely to be deleterious just because it's more likely to mess something up. It's already kind of a gear system that's already in place, so to speak, and more often than not, it's going to simply mess up the workings of that system. So purifying selection is when those deleterious mutations get weeded out, and those regions tend to be less mutated than would be expected if they were neutral, for example.
And normally you're going to find that, well, unless it's a regulatory region, if it's a coding region, it's going to, the things that affect a change in the amino acid are going to be strongly conserved, basically compared to non-synonymous.
Right. Mutation. And it should be, this should be a big point. It should be noted that conservation due to these selective pressures is the similarity emerging from that in the DNA sequence is not necessarily the same as the DNA similarity due to common ancestry or recent common ancestry. You could have similarity in the DNA, for example, because they split relatively recently, two species split relatively recently, and they haven't had much time to diverge in their sequence.
Right.
Or it could be just similar in that sequence because functionally it needs to be that way, sort of, so to speak.
And the real crazy thing is that, okay, if we find something that's really conserved between chimps and humans, but it's not conserved between chimps, humans, and rats,
right?
Then we know that we have something about primates, basically, that is conserved, right? Something in primates.
Right.
And then if we take a look inside of the human population, and we find that there are different geographical regions where humans live, and we find things that are strongly conserved or not conserved. Okay, that's another measure, because they turned out to be human either way. We have humans living on one side of the earth and humans living on the other side, and they have some things that are, that vary quite a bit. So they have mutations that vary, and we know that is a very, very unconserved region of the DNA, basically.
Right.
And probably
because it's so highly variable,
doesn't have a function, so
yeah,
it doesn't have to be a particular way. But then, you look and then you would find differences between humans and chimps. So what makes humans humans? What makes chimps chimps? What makes chimps and humans primates? And of course, we're right into why phylogeny is so fucking important.
Right. Yeah. Well, just to add on to that, like one of those papers you emailed me, actually, I found a graph. I don't know if I can bring it up right now, but basically what it was showing was comparing the amount of DNA that's constrained in a mouse and the same thing in a human. And they both were about 9% of the genome, give or take. Yeah. So I think that's where this number is coming from, because it did kind of separate it between coding and non-coding regions that were constrained.
Yeah, that's very different to what is to a comparison, right?
Right.
And the thing is, what we would expect with splitting 71 million years ago, that's 142 million years of separation between the two branches, right? We would expect that many mutations in these non-functional regions, or even in some of the functional regions where it doesn't matter as much. But we'd expect a lot of neutral mutation between those two. So why do we care anyway that we compare only a little bit? So I mean, the whole discussion, when people do it, like in one of Maddox's, I like to call his late live shows a box of rocks. So, when he puts up one of these boxes of rocks panels, and he has all these people throwing out these little fucking, oh, do you know this and that? And it's like, it isn't that goddamn simple. You got to unpack all that stuff, right?
Yeah. If anyone wants to take away points from this stream today, it's the fact that what Maddox is saying about comparing the genome and getting this nice, simple little number. No one said it was that easy. And on top of that, the number he's citing isn't even what he's proclaiming it to be.
He cherry picked it.
Yeah. And like I was saying backstage, it doesn't even really matter, to be honest, because the argument for common ancestry especially with like humans and chimps, but even for humans, chimps, and mice for that matter, isn't predicated on the exact percentage similarity of the entire genome, because of course genomes are going to vary quite a bit. Right. But what you're looking at is when you compare multiple different regions of the genome, how similar they are with respect to another party, basically to an outgroup, and that's where you get this nice nested hierarchy, and it's that matters, not simply comparing these two sequences. Oh, look, a mouse and a human whole genome comparison, they give this amount of similarity, therefore they're related. Like, no one is arguing that, except maybe people online who don't know any better.
So
sorry, didn't mean to ramble there a little bit, but I wanted to make that point home.
Thank you. We need to drive points home. So, oh gosh, should we talk about the SFT thing first?
If you want.
Get that out of the way. So
I also see we have new people in the side chat. Hello, new people.
Hi, new people. Captive Desk. Is that new?
I don't know. Or I maybe I haven't just seen him in a while.
I've seen Captive Desk, I think. But anyway.
We also have Danish Debater.
I've seen Danish Debater.
Oh, I guess I haven't seen him then.
But I'm not sure how that often. Anyway, so Dapper Dino, Gutsick Gibbon, and Creationists, all three released videos today, which I've linked in my description, all calling out SFT on some of the very erratic and confusing conversations they've had with him.
Oh, it's Dave. Hey, Dave. Oh, sorry. Go on.
Quit changing your name, Dave. So they all, and I watched Erica's. I wasn't aware of the other two until before we went live, but I've linked all three of them. But it was absolutely amazing how like Erica's point was that his behavior was very erratic. It's like, oh yeah, you know, I really appreciate you and I really want to talk to you. And then we try to set something up with him. I had the same result with Cel Cordova over this debate he tried to set up. I mean, he wanted to set up a debate, but then it just kept, he wanted to talk about evolution and abiogenesis in one debate. He wanted to have a 2-hour debate. Then he wanted to have an 8-hour after show.
Oh.
And so he wanted me to be online for 10 hours defending the entire gamut from, you know, the beginning of the earth until now. And
right.
and I finally it finally occurred to me that he didn't really want to debate me. He just wanted to call me out. And that's the same thing with SFT. And he's done this over and over again. He calls us out and said, oh, dodgeball and all that stuff. And then when you try to set up a conversation with him, he's like, oh, I don't want to talk to you, or I don't want to do it on any other channel but my fucking stinking channel, you know?
Right.
Where I have the stink of Hovind lying around. The stink of Hovind is the biggest problem with that channel right now.
Well, I think Dr. Dan also made the comparison between him and a certain DC supervillain, Two-Face.
Oh, yeah? Oh, good. Oh, I'm going to have to watch Dapper Dinos and Dr. Dan.
Which I think is a pretty apt comparison.
Yeah. Yeah, and, you know, I sensed that about him a long time ago, that there was something up. And I think what's up is that in a lot of ways, we are nothing but token fuel for his monetary concerns on his channel.
Yeah.
And I'm pretty sure, I mean, he is a really good YouTube channel producer, and he's very successful. He's becoming successful. And I don't think he gives a shit about much else. Truth be damned. It's just, you know, he's got his ministry. He calls it a ministry. That should be a big fucking clue.
Right.
So anyway, the point is that, you know, do we really need to deal with this joker?
Yeah.
I'm more interested in like Faz Rana and people like that. Yeah.
Actually have a conversation, actually educating people online who want to learn something.
Right, or Paleologos. I think I'm going to have a conversation with Paleologos.
Oh, that'd be awesome.
This week, I think.
Yeah.
Because I got questions for him. He's a real strange one because he accepts it. Well, okay, this is the same thing with Faz Rana. I'm really confused about this what Faz Rana said. Were you confused at all by, like, okay, here's the thing.
It was vague at some points, I will admit.
Well, he's an old
talks
Earth
about being an
creationist.
old Earth creationist, doesn't he?
Yeah. He's an old Earth creationist, I think.
So that means he accepts like a lot of evolution, like possibly even from eukaryotes up to all the animals. He might allow for the special creation of man. So let's leave man out of it entirely, right? So if he believes in evolution all the way from eukaryotes,
why I don't think he believes that.
Well, he said that eukaryotes is one of the big points where he thinks God had to interfere. He did say that in this discussion.
Yeah, but I don't think he's a theistic evolutionist or anything.
Yeah, but he's an old Earth creationist.
Yeah, well, there are differences there. Not all old Earth creationists are theistic evolutionists, and vice versa.
So okay, the point where horses and zebras split. Okay, he would accept that, right? He would accept that horses. Well, of course, all
probably.
Okay, horses and elephants, basically. Would he accept that?
I'd say probably not.
Really?
So yeah, the important thing is I wonder if he sees humans as being totally separate from apes. Like,
yeah, but I can't say anything on his position, only that from what I've seen of old Earth creationists, they don't necessarily accept long-term evolution.
Right. Or that I don't know that they accept a literal. Well, I think they might accept a literal Adam and Eve, as being the actual first human Homo sapiens types. I'm not sure, but
well, we can listen to a little bit.
Yeah,
let's see if we can pin a little bit of this down. I'm not sure. I'm not sure why I bookmarked this, but
this is a great way, for me to really identify, weaknesses in my position and places where we need to spend some time doing, more work. So,
wait, is this a bookmark?
Yeah, that's a bookmark. The reason I bookmarked that is I heard Cel Cordova say the same thing, and a few other creationists when they come on and talk to an atheist, right? Or an evolutionary biologist, they will always say, "Oh yeah, I welcome criticism because I need to strengthen my argument."
Yeah.
Well, wait a minute. What about the truth? Are you worried about your argument and how to strengthen your argument for what you already believe in, or are you worried about finding out new things and finding out if you're right or wrong?
Yeah, that's
you see
true.
the difference?
Yeah,
and I keep hearing that from these guys, and it kind of concerns me, But I don't have any other bookmarks other than the one on pseudogenes, so maybe we'll just run it ahead of here a little bit and see.
I love to have these conversations, so I'm thrilled. I'm honored. I've been looking forward to this.
Let's go ahead.
An agnostic, had no real interest in religious things, and it was just studying biochemistry and becoming aware of what are intractable problems with the origin of life that convinced me at least life's origin, again, required the work of a mind. And I've spent, that was 35 years ago or so, and I've spent the last 3.5 decades continually revisiting that conclusion and written a number of books dealing specifically with the origin of life question. So if there's an area of expertise that I would claim, that would be the area of expertise. And when I look at the history of life on Earth, again, I just see these key transitions in life's history where it doesn't look like current evolutionary theory is sufficient to account for those events. Something I'm becoming increasingly interested in is the origin of eukaryotic cells. That's a fascinating area of science.
Okay, there you go. So he mentioned two things: abiogenesis, where you would need intelligent design, and he mentioned eukaryotic, the transition from prokaryotes to eukaryotes, right? So what is it that he is accepting and what is he denying? It kind of implies that he's a real old Earth creationist and he would accept all of evolution. But then if he does accept evolution, why is he pushing back later on about pseudogenes and ERVs and things like that? Because if you accept a modest amount of common descent between, birds and dinosaurs or whatever, you would accept all of that just automatically. I mean, that would be part of it. You would know that these things happen in genomes, right? You see what I mean?
Yeah.
So
I will say it isn't a consistent position in my opinion, but I like I said, I can't say for certain what he thinks and doesn't think.
Yeah, and that's what I don't think it was ever clarified in all of this.
Yeah,
so
that's true.
I don't know. I'm not sure. So I should have bookmarked more, but well, listen to this. Let's see on pseudogenes. Oh, let's see. Let's start 4011.
I think you're making a strong case or a strong point, but again, I still think we are just so much in our infancy that there's going to be things that are going to surprise all of us.
Okay. Now that's kind of the standard. Oh God, I hate to use this phrase because it's a creationist phrase, but that's a standard talking point.
Yeah.
That, oh my God, we're just in the infancy. We don't understand how complicated all this stuff is. Well,
apparently we've been in the infancy for about 20 or so years now.
Yeah. The thing about that, and we could go over it a little bit and list what this infancy is and what it means. And we are not in the infancy. We are not naive anymore about 4D genomes, about chromosome regions, about epigenetics or any of that. We're not in the infancy anymore. We're at the point where we're sifting the details.
Yeah, I see Orge's inside chat. If he wouldn't mind, I might want to ask him a question or two about rocks and geology, if he's okay with popping in for a little bit later.
I'll pop him a link.
Thanks.
Always good to have the orgy. I like to call him.
He knows his stuff.
I like to call him Orge.
Yeah.
Oh, let's see. Okay. So, I sent a link.
All right. Thanks.
And then let's see. We go on about pseudogenes right here. So.
Right. Oops. Yeah. Well, there was a paper. I ended up writing a blog about it a couple of years ago now, maybe, basically pointing out that the level of transcription among pseudogenes may actually be higher than what we think.
Okay. So I linked his 2 blogs, and I had some server problems. Apparently there's some sort of a virus in some of the blogs, and I reached out to him, and I think they're correcting that. I'd be interested to see if you guys got it or if it's just my firewall that's catching it. But anyway, you guys might get the virus if you pop up. But anyway, I've linked those things, and I've linked at least one of the papers that he was referring to in the description. So I did. I don't know if Dr. Dan did. I think I beat Dr. Dan out in providing good descriptive links. But anyway, so I dug in a little further and looked at some of his blogs. But so he's talking about this paper on. Let's see, which one is it? Here's it, Merle. Let me see here. So basically, there was some confusion as to whether or not we were getting. That's the mouse. Okay, that's Grar. And this is, I think it's the paper's Hertie. No gene in the genome makes sense, except no, I don't think that's it either. And then there's Keitham. Let's see. Overcoming challenges and dogmas to understand the functions of pseudogenes. That's the paper. And I believe I linked that one. And they're talking about how, because they classified them as pseudogenes, they didn't look at them. There wasn't a lot of scrutiny, so they would sometimes rule them out. But now they're ruling them in more. But apparently you can get, well, I think he explains that you can get a false reading where that would indicate that less of the pseudogene, because of the sequence similarity, you wouldn't know that you were actually getting more transcription of the pseudogene than you were getting of the gene, or you were getting or that both of them were being transcribed. So I think that's the point. But that's a pretty weak point. So
because the assays that are performed many times might actually conflate pseudogenes with the intact genes. So in other words, the pseudogenes are being transcribed, but it's being reported as being the intact gene.
Right. It's hard to distinguish between the two.
Right,
yeah.
Right. You know, and again, you know, if they're not being transcribed, that's a problem for my model, right? Unless they're serving some other role, again, that hasn't yet been, you know, defined. I think in this particular paper, and I'm going from memory, so there was some comment to the effect that maybe these untranscribed region or pseudogenes may be playing some kind of role in chromosomal stability. Now, I can't remember how they specifically were making that claim, so I'm going from memory. I probably should have reviewed that before this.
Oh, that's okay. Yeah.
You know,
okay. So if they are being transcribed, and they there's two things that we have to establish. Number one, if they found that they're somehow effective in some sort of stability or stabilizing effect on the genome, I'm going to have to look for that, because that's kind of a stretch. I mean, there are 13,000 pseudogenes.
Right. I
really doubt they're all involved in stability of the genome. And why did the designer make them look so much like a gene?
Right.
That's not functional. I mean, it just doesn't really hold water.
Well, Dr. Rana made the point in the video that they may be similar to genes because they help regulate them. But I think Dr. Dan also made a good comeback that if they're, why do they need to look exactly or somewhat like these genes if they can also not be exactly like them because they mutate so quickly in comparison to other parts of the genome?
Right. So they found one pseudogene.
Which is it? Do they need to be exactly like it, or do they need to be somewhat or not at all like it? Like you can't have it both ways.
Well, and that's the thing. He brought up a second topic. Okay, the first topic is stabilizing a gene. The second topic is they had to be the same because there would be some competitive regulation of the gene, right?
Right.
But I mean, there are like 50 other mechanisms for regulating genes. Why create a whole pseudogene just to regulate another gene?
Right.
Why not create a small miRNA that regulates the gene? You know, if you're designing something, why did God use so many color cranes?
Right.
It just doesn't make sense. Okay, there's a CYP2A7 human cytochrome.
Hey, covalent bond. Nice to see you here again.
Cytochrome P450 enzyme increases CPY2A6 mRNA stability due to a 3' UTR polymorphism formed by gene conversion. Okay, so on the other end of the transcript, we have the 3' UTR. That's always at the very end. There's they call it the 5' UTR at the beginning.
Okay.
So
remind me again, what does UTR stand for?
Untranslated region.
Okay, that makes sense.
Yeah, so you got the 5' UTR in the beginning, and that gets trimmed off before translation. And then at the end, you got the 3' UTR with all those, like a poly A.
Yeah.
There'll be a lot of adenine so that it knows how to handle the gene, basically, and get it out of the nucleus.
Right.
That's the thing. There's a bunch of modifications made. And then, he mentions, so this is in the paper. This is in the Cheatham paper. Cheatham 2020 is the paper. Let's see, are required for suppressing the expression of fetal globin. Okay, another example in which pseudogenes may function intrinsically as DNA elements is by influencing chromosome stability. A study of genomic architecture of the 22q, the chromosome 22, and then they give you the address, locus, which is associated with DiGeorge velocardiofacial syndrome, proposed that deletions of a pseudogene within a low copy repeat region could increase non-allelic, no, there's a hell of a lot of words here, homologous recombination. So they found a function for one pseudogene where if you delete the pseudogene, you end up with some sort of a defect, right? Which is one out. Okay, we got 1 out of 13,000, Mr. Rana or Dr. Rana. That's where he got that from.
Yeah.
And that's
we may disagree with him, but he is an actual doctor, unlike another doctor we've heard of.
Right, yeah. A real doctor. Yeah. Faz Rana is a great guy. Great guy. Yeah. We had him on the channel once. We had a great time. And
you did? When was that?
God, months ago.
Oh, okay.
And he wants to come back, so I think we're going to invite him back and
nice,
and beat him up.
Not too. Never mind.
Anyway, okay. So, but the thing is that all of these things that were okay. You get a copy duplication. The genome changes a little bit. It doesn't kill the organism. It ends up being neutral. It also ends up having some small effect on the entire genome.
Right.
Then some other things change for about the next 1,000 or 100 generations. And then something else locks into place, and all of a sudden it has a function. This is exactly what we expect in evolutionary biology. So all of these things where they're saying, oh, this little thing has function, that little thing has function, that mRNA, that all lends support to the evolutionary theory as a whole. Because this is exactly what we would expect to happen. Because
they're functional, but they have telltale signs that they were co-opted to begin with.
Yeah. And, and of course, some creationists, who I shall not name, would say, oh, show me a paper where a gene becomes functional. It's like, well, how the fuck are you going to do that? That they're bullshitting.
That's not even how mutations are observed to begin with.
Right. Anyway. Okay, so
he who shall not be named.
This was a really good talk with Dr. Rana.
Yeah.
And it just seems like he's stretching and he wasn't clear about where he thinks God intervened or how God intervened. Now, if God intervened between prokaryotes and eukaryotes, I want to know how God intervened. I mean, was there something programmed at the very beginning of the Big Bang that caused that intervention?
Yeah.
Or was there some miraculous intervention? And what would that look like? Would that end up like you would section off 6 acres of ocean and turn those prokaryotes magically into eukaryotes?
Yeah.
I don't think Faz Rana thinks in those simple terms.
Yeah.
I think he has a more complicated model, and I don't think he actually did a very good job of expressing his model, the one he's working on.
To be fair, that wasn't what the topic was about.
Yeah, I guess it was about junk DNA, whether or not. But the thing is,
that would be good to hear what his actual thesis is, I guess.
Yeah, it would. Anyway. So let's see, where's the paper?
You sent Orge a link, didn't you?
I did. I want to make sure I got this paper linked. Oh my goodness, people are texting. And you can see my
text.
You can see my text when I share my screen, and I don't like that.
I wonder if John is lurking somewhere.
Let's see here. Where are we at here? We're at PTFS. Okay. So this is the Cheatham paper, and down here they have a table. They explain pseudogenes really well.
Sorry, Dave just sent you a video. He says he talks about his model here, so I assume he's talking about Dr. Rana.
Good. Okay, so right here we have the functional pseudogenes that we found. So that's the table that we had in 2020. Now, keep in mind that this is 1, 2, 3, 4, 5, 6, about 20 of them out of 13,000.
Wow.
So we got a ways to go. But, as Faz Rana pointed out, we're in the infancy of checking these genes out to find out, and I wouldn't be surprised if they had some effect on biochemistry somewhere.
Yeah.
And I would be less surprised if in 1,000 generations there was another mutation that basically conspired with this one that ended up becoming functional in some manner. And we ended up with an entirely new gene that gave us, 6 fingers to type with, because that would be so much better to do texting and stuff. Or what we need is we need an arm growing out of the side of our neck to hold our iPhone.
Yeah.
Yeah, that's the next step in human evolution.
Yeah.
Okay, let me make sure that I have done this.
Man, that reminds me of all those wacky videos. The next step of human evolution and fins webbing on our hands and whatnot.
Where did my co-host go? My producer, what?
His migraine was getting back up again, so he had to leave.
Oh my God, that's terrible. Okay, let's take a look at this video that Captive Death.
Why not?
Human Origins Workshop.
Man,
Okay.
I really was hoping Orge was going to pop in.
Let's see what we got here.
I guess not.
Because as we've been talking about human origins, and I've noticed this as a trend in the broader science-based conversation about human origin.
Oh.
You're not showing it.
My bad.
There we go.
You're so visual.
There is a lot of emphasis on the molecular anthropology, on human population genetics, now on the genealogies, which I think is really important, of course, but we're neglecting other data sets, or we're de-emphasizing other data sets at times when we do that. So I think it is important to bring archaeological evidence to the conversation as well as evidence from the fossil record. I will also touch very, very briefly on how our model can be influenced by the genealogical atom model. And I'm not going to get into a lot of detail here simply because I'm still in the process of thinking through what Josh has presented, not only today, but in terms of his book, The Genealogical Adam and Eve. And I just want to say that I think Josh has done something really very important, because he's forced us, as a number of the panelists have said, to think about the question of ancestry maybe a little bit differently. And so he's done us a good service in the sense that we've got now another option that we can begin to look at in terms of how can this make our case for human origins much more robust. So we'll leave a lot of that conversation to the time of the panel, to the time of the workshop.
Okay, should we jockey ahead here a little bit or what?
Yeah, Dave said he put up slides if it helps you skip through.
He put up slides if it helps. Okay. Let's see here.
I think he's saying, like, look at the slides, don't listen to the whole thing, maybe.
Yeah, here's some slide evidence for it. Well, let's see what he says.
For lack of a better way to describe it, skeptical about facets of the evolutionary paradigm. And so the question then becomes, well, what do you do with what so many people consider to be the overwhelming evidence for human evolution, like the hominin fossil record, or the shared biological features that we have as human beings with other creatures, including the great apes and the hominins? Well, our approach, and again, I'm not going to spend a lot of time on my presentation with this, is to argue that the hominins were creatures that God created that existed for a period of time that went extinct, that these were creatures that had some intellectual capacity, emotional capacity, they had culture, they had technology, but that they were cognitively inferior to human beings, and that we would see these creatures as creatures that lacked the image of God, that would be the major distinction. And because they lack the image of God, there's going to be cognitive differences and behavioral differences that separate or distinguish humans from the hominins.
You want to comment on that?
I mean, that's an interesting position to hold. It's just, it begs the question, why did he create them, though, to begin with?
Right. Why make them so like us, except without this cognitive ability, that sort of thing? And it's sort of an impossible position to disprove.
Right.
So there's no.
It's sort of like a parent giving a child a car, but not giving them the keys to start it.
Right.
Like, what's the point?
And the thing is that okay, the image of God. So man was made in the image of God, or was God made in the image of man? How are you going to prove one or the other, Mr. Rana,
Right.
Dr. Rana? How are you going to differentiate those things? How are you going to convince me that we didn't create God in the image of man to make man more special on the earth? Because obviously we think we're pretty fucking special, you know?
Yeah.
Because I can outsmart my cats half the time.
Yeah.
Half the time, not all the time.
Cats are pretty smart.
Yeah. Yeah, they come and stick their tails. They probably stick their butts in my mouth while I'm sleeping. I don't even know.
That's a weird image. Thanks
Yeah,
for
that was a weird image. But that's, you know, the cat's ultimate revenge. They do weird shit while you're sleeping. They'll come up and lick your lips or something like that, and that is just fucking just outrageous to wake you up. Yeah. Yeah. And anyway, I digress. Let's go ahead and look and see if we can find some more slides here. Okay.
Is reflecting archetypical designs.
Okay, I linked his blog post on Owens, I think it was. Pre-Darwin.
Richard Owen.
Yeah, he came up with this vertebrate archetypes, which is really forward-thinking. But vertebrate archetypes, when you look into embryology, that's what it's all about. When you look into vertebrate embryology.
Owen was critical in elaborating the concept of homology, and prior, again, to Darwin, he understood these as reflecting archetypical designs that originated in Owen's thinking from the mind of God. That was, and those.
He looks like Scrooge.
Yeah, it kind of does, yeah. At least later in his life.
I think it is Scrooge.
Okay. He acted like it sometimes, that's for sure.
Within the created order. Now, so our approach, again, to shared features is common design, not common descent. Now, here's a point, and I'd be interested in Josh's reaction to this. There are some elements, I think, of the genealogical Adam model that actually employ common design. So if one version of the genealogical Adam and Eve model would be that Adam and Eve were de novo creations in the garden, and that they were created to be biologically compatible with the humans outside the garden that evolved. And if that's the case, then this is...
Okay. Did you get that?
No, not really.
Okay, let's listen to that again.
Interested in Josh's reaction to this. There are some elements, I think, of the genealogical Adam model that actually employ common design. So if one version of the genealogical Adam and Eve model would be that Adam and Eve were de novo creations in the garden, and that they were created to be biologically compatible with the humans outside the garden that evolved. And if that's the case, then this is...
what?
Yeah.
Yeah, that doesn't... wait. Okay, I don't even understand what he's talking about right now, but that's probably just on me.
Okay, I don't know if that's his position, but it's one way to accept all of to accept primate evolution. Okay, that there were all these humans that evolved outside of the garden from primates, and then God did a de novo creation of Adam and Eve with cognitive.
Wait, so he's saying we were created to be compatible with other primates?
I don't know if this is Dr. Rana's position. I think he might be talking about Josh.
Yeah.
But yeah, so we were created to look like the primates that evolved outside of the garden, but at the same time, we were created in the image of God and given this cognitive ability. So, yeah, this kind of sums it up. But I mean, it is... You know, I'm okay if they come up with a really good model like that. Fine, as long as they don't deny any of evolution.
Right.
I'm fine.
They don't establish science.
Yeah, and I kind of have hope for Dr. Rana that he is working on that sort of a model. But you know, but then in Dr. Dan's video, he sort of broke my heart because he kind of went against it a lot.
Yeah.
So.
But it isn't to say it wasn't a good conversation either way.
Still broke my heart.
Yeah.
Fredo, you broke my heart. Anyway, okay. Let's see what he says.
Example of common
Midnight
design.
wants in.
He wants what?
Midnight wants in.
What in the hell is going on right now? Oh no.
Can you hear me?
Oh no, hang on. I have to mute.
Oh. So folks, hope you are enjoying so far. Looks like I've officially taken over Answers and Atheists. Never mind.
I'm back. My watch started playing a song. I have no fucking idea why.
That's weird. Huh.
Yeah.
I was going to say, I have officially taken over Answers and Atheism Incorporated, but you stopped me short of it.
See? Mutiny stopped. Mutiny over.
Yeah, I wasn't saying it was a mutiny.
Take him away and hang him.
Oh,
wait a minute. Scott's not here. Guess you are in charge. Okay.
Should we put the link out? Maybe some people want to join at this point.
Yeah, I'm just about done for the day. So I want to warn everybody that this is going to get shot in the head shortly. So a coup.
A coup.
Yeah, I like coupe. It's a coupe.
The chicken coupe.
A coupe. Oh my God. Reminds me of my childhood. Clean chicken coop. Oh, don't go there. Okay.
Where God is creating with common design. Just an interesting point of connection. And again, I'd be interested in seeing Josh's reaction to that point. Something that was, I think is really very important, as Josh has driven home very well, I think, is the idea of defining what we mean by human. And that was one of my favorite chapters in the book that Josh wrote, is the chapter on...
That is a good question.
The definition of human.
Or who's he talking about, Josh? Swamidas?
Yeah, I think someone in chat said it was Swamidas.
Good morning, good evening, and good night, Mr. Andrew. And
hello.
Answers in Atheism. How are we doing?
Good midnight.
We are upside down.
Are we?
I don't know. I'm just I'm saying something random.
Oh, the upside down world in Stranger Things.
Andrew, you are the most you are the most random person I've ever met in my life.
Oh, thanks. That's flattering.
Hey, Speed.
Yeah.
Shoot it in the head.
Pretty quick. We're doing
okay,
some
yeah,
science.
Apparently it was Swamidas.
We're doing science here.
Science doesn't exist.
Don't interrupt us.
All right, fine.
Were you on Rebecca's stream last night?
A little bit, yes.
Okay.
Oh, Rebecca streamed last night?
Yeah, she had some interesting discussions with who is okay. Rob has some pretty radical views on Christianity, right?
Oh, is it another Christianity stream?
It was a Christianity stream, yeah. But
got my hopes up.
Did you get that midnight that Rob is pretty radical and he got a lot of flack from the other Christians?
I'm not surprised.
Anyway,
we did.
Yeah. I want to review that, but I kind of don't care about Christianity.
Do you care about Rebecca?
I do care about Rebecca, but
then do it for her.
Well, I'm interested in Rob, actually. I'm interested in a guy with views that, cross over. I'm looking for the bridge. Anyway, that's a whole other discussion. Let's go ahead and see what this guy says here.
Or the chapters on the definition of human. And of course, scientifically, you could think of human as referring to hominins, so that would include the Australopithecines and related genera like Ardipithecus and Sahelanthropus, as well as the habilines and the erectines and archaic Homo sapiens. Or you can take human to refer to members of the Homo genus. So this is how scientifically the idea of human would be defined. But what we do at Reasons to Believe is take the notion that human essentially would refer to a Homo sapiens sapiens, to anatomically and behaviorally modern humans. That's what we mean by that, but that these humans, again, it would be distinct from other creatures in that uniquely Homo sapiens sapiens, or anatomically and behaviorally modern humans, possess this quality, again, called the image of God. Now, it's interesting that when you look at thinkers, scholars who are working in the science.
So I don't know. This is just a weird way to phrase it. The image of God is a quality. I don't know. That just sounds weird.
So this relates to this whole thing about consciousness, that humans are conscious and animals aren't, and we have this special additional quality.
Yeah, well, I definitely wouldn't say we're the only things out there that are conscious.
I wouldn't either, but we are the only things that can come up with really abstract. I'm not even sure about that.
I'd say it's a spectrum. It's a
spectrum,
and we're closer to the one of the ends of it.
But there's something really interesting about the human brain. Okay, inside of your brain, you have something called the fourth ventricle, which is a big kind of a big ball of water, right?
Yeah.
That sort of protects your brain and nourishes it and stuff. And at the front of the fourth ventricle is something called the caudate nucleus, right? And in humans, as opposed to primates or any other mammals, there is a swelling of some extra, it looks like some extra pathways that got developed that interconnect your lower or the basal parts of your brain with your cortex. So it kind of looks like we are capable of creating one more loop than other animals. So we evolved a second loop so that we can do things like think about thinking about thinking.
Right.
And we can use that loop basically regressively, infinite regress, so that we can construct concepts about concepts about concepts. Right. And that seems to be there's a possibility that we do have an extracortical loop, and they've also correlated that with IQ differences among human beings as to how much of that loop you have. But that's kind of a bad road to go down for humanity.
No.
You're a bad road to go down for humanity. I don't know.
Well, I am. Yeah, I don't know why you would associate with me, Midnight. you are monkish and pure, of pure heart. I don't know why you would hang out with me. But anyway, I'm going to influence you. Let's see what Fuzz says.
With the Force.
Faz Rana.
The Force, yes.
Who have
the Force of darkness.
Are seeking to identify who Adam was. There's a number of options that I've seen people explore. One is to say that maybe Adam was an erectine, one of the species that would belong.
Yeah, okay. So apparently he's going to talk about that. What else did he talk about?
I'd say an interesting position, that's for sure.
Yeah, that's an interesting video. I did link it, so I'm not sure. If maybe it will, maybe I'll get some timestamps and we'll spend a little more time on it in a later show.
Yeah, when we're more prepared.
Yeah, but and that is the basic problem I have with Christianity, is there's a lack of humility, that they actually think that we are this one special species.
Yeah, I tend to agree to some extent.
Well, it ends up being a destructive position even for our own humanity.
That's true.
Because it invites a selfish, I want to live forever, I'm special sort of thing. And Midnight is like he's very typical of that I want to live forever mindset. So I'm glad you
wait. Okay, here's the thing. I believe in evolution and the Big Bang and all that thing, right? But for me, the fact that those things happen is what actually makes us special, not the fact that we were created by, not that we were created by some magical sky wizard or whatever the hell, right? To me, what makes humans special is the science, is the natural explanation. So
let's I don't know, just for argument's sake here, let's take it back a few steps. What were the chances that your mom and dad came together in the right combination of genes to make you? What are the chances that so on and so forth, their parents down the line, and that our ancestors survived all the extinction events, that life got started, that the planet got started? I mean, you can go back all these steps, and I agree, it's I mean, it's probably extreme to say it's a miracle because, again, we don't know everything about the details at this point to say if it was a statistical anomaly or not. But I definitely say that makes it worth living, right?
Yeah.
Yep.
Yeah, the fact that you're alive, but then, this whole idea that, oh God, I want to live forever. What exactly do you want to preserve? Your individual memories, which, like, if you're as old as I am, your memories of your childhood start to get pretty fucking weak and pretty few and far between.
Yeah.
And so okay, your personality.
Do you really want to have this discussion with me here? Because
Yeah.
you know how I feel about immortality.
Your personality. I mean, do you think that midnight is something that we need to preserve for the next 60 trillion years? I mean,
yes.
Why?
Because I'm special.
Oh, yeah, but what if you turn into because you're going to change the whole time. What if you turn into a conservative yak or a dick or something like that?
I mean, what if I turn into a conservative yak now? Like, what's
Are
to
you
stop
guys
me
talking
from?
about reincarnation? What's going on
No,
here?
immortality.
Yeah, immortality. He wants to live forever. It's like, what do you want? Like, okay, I've done a lot of really hard work educating myself. And the only thing that bothers me about my death is that's going to get flushed down the toilet in about 2 minutes, right? That bothers me, but then am I that fucking special? My particular education and view on things? I mean, what is it that I want to preserve forever?
That's a good question.
Speed. You're only as special as you see as you see yourself to be, right?
Oh.
So it's like, okay, you could ask, am I that special?
So
right?
your argument is that if you're a complete narcissist, then you're more special than.
Okay, speed. Let me tell you a little something here. Everybody's a narcissist. Okay, it just depends where on the spectrum you fall. Deep
That's
narcissists.
Right.
Thank you. Deep narcissists are the ones who we usually think about when we hear the word narcissist. They are the ones who, they're the ones who have no sense of self, and they project their own failings onto others. This is known as self objects, right? Functional narcissists are the people who who have a healthy sense of self, and they just go about their business. They do whatever. They really don't care. Healthy narcissists, or those who have a healthy sense of self, but can also put themselves in other people's shoes. Those will be known as the empaths, right?
So those are the guys that are special enough that. So, I mean, your argument is that if you're a narcissistic empath, then you deserve to be around. Otherwise, fuck you.
I mean, that's not my argument, but
I know.
Kind of. Not really.
Sort of straw man you a little bit.
Yes.
Yeah. Well, that's my job.
Okay. Wizard, nobody is special. Boom.
So now I have been
told. Okay, Tyler Durden.
I have been told that around the 13-minute mark here, we have. We gotta get rid of Danny. Hi, Danny. We got to get rid. Let's see.
Yeah, I do like that. I don't know if Gur was intentionally making that little
contradiction.
Yeah, but that was kind of funny.
Well,
no offense, Midnight.
Gur always. Wait. Okay, hold on a sec. I always Midnight always brightens up the day, but then Danny says that's a contradiction. How is that a contradiction? I always brighten up the day.
Midnight is dark and brighten up the day, and
you're a walking contradiction, Midnight. Yeah.
I mean, I am a freak of nature according to some people, so I get it.
Well, that's kind of harsh.
Yeah, okay. Well.
So I think Dr. Rana explains his position here at around timestamp 13. So I'm just going to let it run and see if we can figure it out.
Lacked the image of God, that would be the major distinction. And because they lack the image of God, there's going to be cognitive differences and behavioral differences that separate or distinguish humans from the hominins. And we would argue
just pause real quick.
Created by God.
Okay, now I'm confused. Is
Yeah, that is
cognitive
kind of retarded.
processes the image of God, or is the image of God what led to the cognitive superior?
I don't know. Yeah,
that is kind of confusing.
Yeah, the creator God. So, okay. But when you think about this, what he's talking about is we talk about designing things. Well, designing things doesn't amount to diddly squat unless you go out and build things. And we have these opposable thumbs and we have these fine, intricate hands, and fingers. And we are a tool making species. And that's so we think about everything as creating and tool making and building a house and going out and
we're basically a puddle. We're basically a puddle who thinks that the universe is built for itself.
Something like that. But you see what I mean, Andrew? The anthropomorphic idea is based on a tool making animal, an animal that goes out and makes New York City, Manhattan, right? Things like that.
Well, also an animal that's able to communicate and speak, because I mean, elephants, for example, they can make sort of kind of tools, or chimps too, and dolphins. But since they
I mean, how do you know?
It's a tricky subject for sure.
How do you know? How do you know? How do you know they don't speak as well in their own?
Yeah,
language?
that's why I'm saying it's a tricky subject.
But you got to admit that Manhattan is more impressive than a beaver dam.
That's true.
So we are very.
No. Manhattan is more impressive than Beaverhattan.
Well, I mean, from a termite's perspective, maybe a giant termite nest is like the size of a skyscraper.
Yeah, exactly.
So, like, isn't that impressive from a termite's perspective?
Yeah, but that's the thing. We're full of ourselves. It's because we built Manhattan and fucked that island up.
Yeah.
We're really impressed with ourselves. So we think, oh God, if there's a God, he must be just like us. He built a Manhattan, except his Manhattan was the universe.
Or to give another example, obviously creationists will disagree with this, but the fact that mere bacteria polluted the atmosphere with oxygen and enabled everything else to be possible.
Oh, they did more than we did, didn't they?
I mean,
I'm just giving that example.
Would you really call it polluting the atmosphere?
Mm-hmm.
I'm using poetic language here.
Yeah, oxygen is pretty fucking hazardous.
It is pretty dangerous, that's for sure.
Yeah. Okay, let's see what he says.
I would argue that minimum that creation would consist of creating the major groups of hominins like the Australopithecines, the Habilines, the Erectines, the archaic Homo sapiens. But within those groups, we would allow for the possibility that there could be, evolutionary events, speciation and things like that could create some limited diversity within those groups. Our approach is not ad hoc. If you look at the history of thought in biology, there were prominent scientists that would hold a view of the fossil record very similar to ours. One would be Louis Agassiz, the Swedish geologist, who argued that the history of life reflects a progressive series of creation events with biological groups originating from the mind of God. And so we would follow in Agassiz's footsteps.
Okay.
Well, I mean, it is true that they said that, but that doesn't necessarily make it correct. Just saying.
Yeah, God, he isn't much different from a young Earth creationist in that he believes that God intervened at each group level and created these groups, and then they diversified. So he's talking about created kinds.
Yeah, a little bit. Yeah.
So I mean, I think that probably sums it up. But I'm going to listen to this whole video in my my own mind, and we'll see what happens. So, Dessel, you're so wrong, Dessel.
Okay, what did Dessel say?
Making creation. My
Making
goodness.
creationism look
I
good.
think is he talking about your nihilism or something?
Yeah. Okay. I guess he does remember when I had my religious experience.
Or maybe he's talking about your scientism, or I don't know.
I'm not sure. I don't know.
Why must the good die young?
Because
why must the young die good? I don't know.
Whatever.
Whatever.
Whatever to you, Jack.
So who's watched Stranger Things?
I got a better question. Who's watched the TV show Forever?
Not me.
I think I did, but I can't remember.
I don't watch TV that much.
I am Harry Morgan, and I'm immortal.
You gotta watch a little bit.
What was that?
You gotta watch a little bit. So I heard that the next move by the Supreme Court, I think, is they want to allow states to go back where the Senate, basically the Senate in the individual state can pick who got elected. Now, isn't that going to be fun?
I mean, why don't we just get rid of the fucking federal government altogether and become like 50 countries?
Well, why don't we just get rid of voting and let a bunch of people sitting in all these different state capitals decide for us who's going to lead the country?
Yep.
That's really on the table. They're really talking about shit like that. And I guess originally, they're called originalists. I guess originally that's kind of the way it was. They didn't consider the average farmer capable of electing, deciding who was going to be.
Yeah, that's true.
And so they kind of want to go back to that. And are we ready to go back to that, America?
I don't know.
Are we ready to give up our rights to elect a president? Something we all ought to think about in November when we all get our asses to the poll and vote a straight Democratic ticket. So
today we lose our votes. Tomorrow we lose our ability to eat ice cream freely.
That might happen.
Perish the thought.
Yeah. So who's that guy? Is it Putin or Putin?
Putin.
I like Putin. Putin might die, and if Putin dies, maybe there'll be a revolution in Russia, and Russia will have a true democracy, and we can all fucking.
Wait, how would Putin die?
He's not looking so good. I don't know how old he is, but he's not looking good, and there've been some leaks about health concerns. So I don't know, but maybe
Mere America propaganda. Russian president is strong as ox.
Oh yeah. Ride horse without shirt. I guess that's Trump is planning on doing an ad like that where he takes his shirt off. So, but it hasn't. They're having trouble. They want to rate it like NC-17 or something because of his man boobs. Anyway, it's just what I heard.
We should probably just leave it at that.
We should. We should shoot it in the head. Okay, guys, we're gonna kill it for the day. I am so sorry.
Get it with fire.
We're gonna kill it. Okay, we're gonna have to kill it. Okay. So yeah, we're all done for the day. I linked a whole bunch of stuff in the description. A lot of videos, everything we talked about, some papers. Please look at my description, or I will lose hope.
Yep.
Okay.
You lost hope. You lost hope a long time ago, Speed.
Andrew, why did I let this fucker in here? Why did I let Midnight come in?
Because I love you.
I said to let him in.
Okay.
That's who.
Okay. Anyway. Okay, we're shooting in the head. Goodbye, cruel world.
Bye.